both will be having same light harvesting complex, same, these both will be having LHC, light harvesting complex. But what is the difference? These both will be having reaction center, but the reaction center of the PS1 photosystem 1 will be called the P700, and the reaction center of the PS2 will be called the P680. Why these both name are given? Because this one photosystem 1 capture the 700 nm light of the sunlight, and this captures the 680 nm light. That's why these both are named as the P700, P680. But all structure will be the same, but only difference of the P, which is a light capturing. After that, here that I told that this electron will be captured by the primary electron acceptor. So that name is the phyophytin. Actual name is the phyophytin. So that electron, when it will get excited, it will be captured by the photoelectron acceptor, primary electron acceptor. Now what will happen? Now here I am making here we are having thylakoid lamella. This is called the thylakoid lamella. Here photosystem 2, photosystem 1. Always first will be the photosystem 2 and second will be the photosystem 1. As I told here, this was also photosystem. So here this is the reaction center. When electron will get excited and captured by the primary electron acceptor, so then this electron will be moved here towards PQ, then side B67, then towards the PC, right? Now what? These are PQ, P6. Like this will be captured by the primary electron acceptor. Then this electron will move towards the PQ. What is PQ now? PQ is palestocyanine. This is palestocyanine. We can say these are the chemicals, right? Where electrons from this PEF will pass to the PQ palestocyanine. Then this electron will pass towards the site. This is the site B6F. That is the cytochrome B6F. This is cytochrome. These all are, we can say, chemicals from which the electron is passed. Then it will pass to cytochrome. Then this electron will be transferred to the PC. Now what is PC? PC is palestocyanine. Then it will pass to the palestocyanine, right? Now here, like these all electrons move towards this one photoelectrode. This is captured by the primary electron acceptor. Now to here, to the chlorophyll A, there are no electrons present. So here one H2 molecule will break and it will make hydrogen ion and oxygen and electron. So these electron will move towards the chlorophyll A. Basically this is a thylakoid lamella that is present in mid between the thylakoid. So the one H2 molecule will be we can say break down, and the electron that is converted from the H2O, that will be moved towards the chlorophyll A, because there is the deficiency of the electron. These all electrons are captured by this and this cycle will occur. So there will be no electrons, so that these electrons will move towards there. So and the oxygen will be released in the atmosphere, as we all know that plants give us the oxygen, so that is released by here, this process that is released. So oxygen will be released in the atmosphere, and the only hydrogen ion will be remain, right? So here electrons happen, same cycle will occur here. After that, here what will occur, here also electron present, this is a photosystem too, right? So the electron will get excited and captured by here, also the photo electron, primary electron acceptor present, so that will be captured by here, and then this electron will move towards the FD. This is FD, right? What is FD? This is a ferredoxin. This electron will move to the ferredoxin. After that, now here, as before I told, in light reaction we will make two products, that one was the ADP and one was the NADPH2. So here first, now here basically this was thylakoid, right? Here stroma is present, as in the structure I told, this is a stroma. Here stroma is present on the side of the thylakoid. So this FD, now in stroma we are having NADP. In stroma, NADP are present and many hydrogen ions are present, right? Now this electron will move here. For the making of the NADPH, what is needed? NADP plus electron plus hydrogen ion plus NAD plus NADP synthetase. This is enzyme needed for here. Basically, NADP synthetase is present here on the outer side of the thylakoid. Here NADP synthetase is present. This is enzyme present on the outer side of the here, on the outer side of the thylakoid. Now for the making the NADPH, we need the NADP that is present in the stroma, hydrogen ion that is present in the stroma, and the electron that is captured by the FD that will be moved towards the stroma for the making of the formation of the NADPH. Then NADP synthetase that is present on the outer side of the thylakoid. Now NADPH is formed. NADPH is formed. Now here we can say the presence of the ions, hydrogen ions is less, but the presence of hydrogen ions in this is more, in this thylakoid lamella is more. For the formation of the ATP, we need more hydrogen ions. So in the comparison, if we do comparison in the thylakoid lamella and in the stroma, we need more hydrogen ion in this, so we use hydrogen ion in the making of the NADPH, because when we do comparison in this, we need more H ions, we can say more hydrogen ions in the lamella for the formation of the ATP. Now here the NADPH is formed. Now stroma, many hydrogen ions are present, right? This one PQ. Now when this electron move towards the PQ, so that it is having more energy, so it will capture the hydrogen ion from the stroma. Because it's having energy, it is having electron that is captured from the PEA, so it will capture the hydrogen ion and it will move towards the CYT. Hydrogen ion will pass to the CYT, and this CYT will pass this hydrogen ion in the lamella. Now you can see there's a more, we can say, the formation of the hydrogen ion. Here I forgot to tell you one point, that when H2 will be break into the hydrogen ion and into the oxygen and electron, this process is called the photolysis. What is photolysis? What is photolysis? When the hydrogen ion will be break into the water molecule will break into a hydrogen ion plus oxygen plus electron. That is called the photolith. What is mean by the photolith? Photo means in the presence of the light. In the presence of the light, and lysis means breakdown. Breakdown. So that is the photolysis of the water. Photolysis of the water. So the breakdown of the water in the presence of the light. So that is called the photolysis. So here the photolysis process will occur here. But one important thing that I am telling, that the photolysis will only proceed by the photosystem 2, not by the photosystem 1. How? Because when this is the photosystem 1, right? The photosystem 1, when the electron will deficient, this was deficiency of electron by the photosynthesis 2. So that will be proceed, this photolysis will proceed by the photosystem 1. Here we didn't need these electrons. These electrons will move toward here, not here. So this photolysis will be proceed by the photosystem 2, right? After that, now what will occur? Here NADPH is formed. Now here we are having one, we can say, particle called the F0 and F1 particle. F0 and F1 particle, that by which this hydrogen ions, these are the hydrogen ions, right? This brother is a stroma. These are the hydrogen ions that will move fast by this, fast by the F1 F0 particle. So in stroma we are having NADP, right? NADP and phosphate. Now in this, now it is having energy, right? So by this energy, this NADP and phosphate will be converted into, not NADP, ADP. ADP plus phosphate will be converted into the ATP. How? When these hydrogen molecules, we can say hydrogen ions, will move by this, it is will having the energy, right? So when the energy will be here, so the ADP plus phosphate will be we can say converted into the ATP by the help of the this one hydrogen ion energy. Now you can see here why we was making the hydrogen ions, only because of the formation of the ATP. When these hydrogen ions will move by this, it will be having energy. So this ADP and phosphate present in the stroma, so these will be these will be converted into the ATP. Now formation of ATP and the NADPH, these two products are the products of the light reaction, you can see here.